Hello students, now let us discuss the general structure of RNA in this module. Once again, look, listen and learn. Now, if you look at structure of RNA, how did you see it in the previous module? Just like this. See that? It's nice and simple structure of RNA. Unlike DNA, RNA is a single-stranded molecule. Right? And how does it appear? Either it appears like this structure right here, which is simple and straight, or sometimes the RNA could also appear like this structure, where it may coil upon itself. And this type of RNA is simply called as nothing but folded or coiled RNA. Is that okay? It has a tendency to fold or coil upon itself. Is that okay? Moving on. Remember, RNA also chemically is a polynucleotide, which means it is made up of several nucleotides attached end to end. Let us look at a single nucleotide first, and then let us see how it is a polynucleotide. So I'm going to again show you a recap of nucleotide. A nucleotide will have what? A sugar, right? What sugar is this? You guessed it right, a ribose sugar. Ribose sugar is a pentose sugar, so let's label the 5 carbons. You see, 1 prime, 2 prime, 3 prime, 4 prime and 5 prime are the 5 carbons of this pentose sugar. At the first carbon is attached a nitrogen base. So what you see here is a nitrogen base. And on the 5th carbon, you will basically see a phosphate group. So what you see at the 5th carbon is a phosphate group. And this is one nucleotide of RNA. And because it is a polynucleotide, I'm going to add some more nucleotides here. Let's go ahead and do that. You have another nucleotide. You have yet another nucleotide, and finally you have one more nucleotide. What I've tried to show you are the 4 different nucleotides you will see in an RNA molecule. Now closely look. On the top you have A and G. Now A and G are the purines that are found in RNA. At the bottom you have U and C. U and C are the pyrimidines that are found in RNA, right? We already know what they stand for. Yes? Cool. Moving on. Now carefully see here. Okay? At the bottom, I've also labeled the 5 carbons. Look at the bottom ribose sugar. Look at that position number 3, right? What do you see? A free OH group. So I'm going to label that end as the 3' end. And look at the top. What do you see? A free phosphate group on position 5. I'm going to call that as the 5' end. So a typical RNA, which is single-stranded, would also have a 3' and a 5' end. Now these nucleotides are going to be joined by the special bond or a special linkage. And what do we give it a name? The name is, you get it right, phosphodiester linkage. So just like DNA, even in RNA, the nucleotides are joined together by strong covalent linkage called as phosphodiester linkage. Is that okay? Moving on, look at this structure of RNA. This is the one that is folded upon itself. So in the RNA, if coiling or folding occurs, then you will see base pairing. And this time, what will be the base pairing? You guessed it right. A U will pair with an A, and an A will pair with a U. And you know that between the two, there will be 2 hydrogen bonds. I have indicated in the figure here. And obviously, the G will pair with the C, and the C will pair with the G, and with the head of 3 hydrogen bonds, right? So it is very similar to DNA. The only difference is here you will see uracil and not thymine, which is going to pair with adenine. Is that okay? Now this is RNA, usually single-stranded or folded upon itself. So I can also say that in RNA, the base pairing is not fixed and is not definite. Is that okay? And because of this, you should also know that in RNA, the ratio between purine and pyrimidine may or may not be 1:1. Right? Because it is a single-stranded molecule. In DNA, it is always 1:1, but in RNA, it may or may not be 1:1. Is that okay? So in this module, we talked about the general structure of RNA, And in the next module, I'll be talking about the type of RNA, where I'll be discussing with you mRNA or messenger RNA. Thank you and take care.
Ini kan biar control ini.